In these images, you see flowers. Obviously, the flowers possess nectar, which has sugar, which we would taste as sweet. The leaves of these flowers, however, are poisonous to us, or at least they contain toxins, and they would taste bitter to us. Obviously, our taste buds and the G protein coupled receptors, which serve as receptors for taste, give us many different types of signals about things in the environment. There are four primary tastes that humans perceive. The ability to detect sweet taste is adaptive since it allows the body to identify foods which contain the potential energy stored in sugars. Sensitivity to salt is important because it encourages the ingestion of foods which can replace the ions which we may lose through sweat. Ions are important for the nervous and muscular systems to function, and so salty foods allow us to replace these ions. Sour foods contain acids, perhaps because the food has decomposed. Such older decomposed foods may contain bacterial toxins, potential parasites, etc., and sour tastes are perceived as unpleasant. The ability of the human tongue to detect bitter compounds is far more sensitive than any of the other tastes. Many of the compounds which are perceived as bitter are poisons if ingested in sufficient quantities. Because of the unpleasant perception of these compounds, humans are unlikely to ingest sufficient quantities to be poisoned. Such poisonous compounds are quite common in the natural world, given that plants commonly synthesize biologically harmful substances in their leaves as a deterrent to insects and herbivores. The family of G protein coupled receptors, which is responsible for taste, can be subdivided into subfamilies. One subfamily, the T1R family, is involved in the perception of sweet tastes from sucrose, monosaccharides, artificial sweeteners, and certain amino acids such as tryptophan, glycine, monosodium glutamate, and others. Because of the importance of identifying compounds which are poisonous to us, we think over 100 genes of a separate family, the T2R family, which is involved in perceiving bitter tastes. Now, even though they respond to quite different chemical compounds, they all result in the same sensation as bitter, which we tend to reject, and different taste buds might actually express different taste receptor genes for bitter so that different compounds can stimulate the same cell. Many of these are located in clusters on specific chromosomes. So for example, there is a cluster on chromosome 12 and a cluster of genes on chromosome 7 of T2R bitter receptors.